Hello, and welcome to Beyond Biotech, the weekly podcast from Labiotech. I'm Dylan Kassane, and this is episode 198 for the podcast. Today, I'm welcoming two guests. Quinn Wills, CEO of Ochre Bio, a biotech developing RNA therapies for chronic liver disease using AI models, and Stefan Baj, CEO of Lexogen, an RNA transcriptomics company and NGS service provider. It's a deep dive into cutting-edge transcriptomics, human-first data, and artificial intelligence. I hope you enjoy our discussion. Quinn Wills, welcome to Beyond Biotech.
Thank you very much. It's an absolute pleasure to be here.
Stefan, welcome to Beyond Biotech as well.
Hi, thank you very much.
Look, I want to start somewhere a little bit bigger than just either of your companies, because I think that's really the right place to begin. Let me set the scene for you here. Drug discovery, it's had a productivity problem for decades. Costs keep going up, failure rates in the late stage trials are just really stubbornly high. And a significant part of that failure comes down to the fact that the biology being studied in the lab isn't human. Quinn, let me start with you. You built Ochre Bio on a conviction that this really has to change. Tell me about the problem you set out to solve and why you think this different approach is possible now when it hasn't been before.
Absolutely. Happy to. So perhaps a little bit of a backstory on why liver disease. I'm originally a clinician before I turned into an AI type. And way back when, we're talking about 25 years ago, I really got to think about liver disease when I was studying alcoholism back in South Africa. We have, I might have mentioned this before in other pieces, but in South Africa, under apartheid, we had this DOP system where you could pay people in alcohol. And it created a very sad situation where alcoholic liver disease is a massive problem in certain parts of South Africa. And that's sort of how I got into thinking a lot about liver. And fast forward 25 years, really, it still is such a big passion of mine. It is, for me, the two key numbers to know here are 1.5 million people globally a year die of liver disease. And really, for the vast majority of those folks, the only real curative option is a liver transplant. And if you think that only about 40,000 liver transplants get done globally a year, it's a health lottery. It's not even healthcare. We're not even close to healthcare with liver disease. So it's a huge issue. And it's an issue that disproportionately affects disadvantaged communities, as we've seen in South Africa. So that's really been the mission. It's been a big mission in my life. But there's a problem beyond just the patient problem. And the problem is how we make therapies in this space. And there's a bigger problem, as you've rightly addressed. And then there's also just the micro problems that we see in developing drugs in the liver space. And we boil it down to three things as a team. And Ochre as a company is really founded around those three ideas. First is, we really just don't have enough causal human biology knowledge around the liver. It's been said that we know more about the human brain than we know about the human liver, and I think that's probably true. And while we've had some successes with genome-wide association studies and the genetics around liver, there really isn't much out there to discover new targets in the same way that we perhaps have in other organ systems. So that's problem number one, that there's already your first human data problem. Problem number two is the models to validate ideas around targets are not really great. The gold standard mouse models still don't really predict fibrosis regression, for example, in fibrotic liver disease, which is ultimately cirrhosis and a big challenge for us in the space. So there's that. And again, we'll get into the human data problem, the human model problem around that. And then the clinical trials, this is the third big bottleneck in our space, are really challenging. Liver trials are of the most expensive, high-risk, challenging trials. We've had successes, but a lot of it again boils down to a human data problem, how we think about human data, how difficult it is to get reliable, good human data in clinical trials. So it's really addressing all three of those, but I imagine today's conversation is a lot around the first elements of that. It's human discovery data and human validation data.
Stefan, if I come to you, this drug discovery is relying more and more on this NGS data. How is Lexogen supporting clients in the field?
Well, I mean, we clearly see that more and more teams are using RNA sequencing data in order to better understand how diseases and treatments affect gene activity in human body. This is clearly a trend. And transcriptomics actually really gives an advantage over technologies because you can really see the broad and have an objective view of what is happening into the cell. And this is really helping researchers to discover new biomarkers, to identify new drug targets, or even to better understand diseases. And the idea actually is very simple. If you start with better data, you can make better decisions later. And this is what Ochre has done. It also allows people to reuse the data and to continue learning from this data over time. And this is the essence of the perturbation atlas, which has been done by Ochre. So at Lexogen, we've been working in the field of RNA sequencing for almost 20 years now. So we have basically grown with it. And even 15 years ago, we have pioneered the 3' RNA sequencing with our QuantSeq technology, which is especially well suited for companies or for teams which want to study the gene expression and compare the expression of the genes according to different conditions. So as a kit maker, because we are also a kit maker, we continue to develop new tools, innovations in order to support drug discovery. But also as a service provider, we try also to be a bit different from competitors, because what makes us unique is that we understand that every single project has its own requirement. Every single project is unique. So what we do, we first take the time to really understand what our partners really need, what they want to achieve, and what kind of material they are starting with, because it's very important. It varies a lot if you are starting with hepatocytes or with cardiac cells. And then we are adapting the workflow, we are adapting the technology, and finally, we also adapt the data generation in order to make sure that we are really supporting the goals of our partners.
Lexogen has developed into a trusted RNA sequencing supplier and NGS partner for drug discovery, but it's been in the field for quite a while. In your opinion, Stefan, what have been the major developments in transcriptomics over that 20 years?
Well, you know, Dylan, I mean, this field is really exciting. It's really exciting because it's moving incredibly fast. I mean, every year there are new technologies which appear. The existing ones are getting better. And now, and if you have been attending some of the main congresses since the beginning of the year, you see that we can see the biology in much more details than before. You can combine different types of data to get more complete picture. And nowadays, spatial and multi-omics are on the lips of everyone. It's really interesting. And another big change is the fact that sequencing has become easier to access and more affordable. So today, not only the large companies or the academic labs which are well-funded can access it, but also the small lab can use it, which is really good for innovation. However, I would say that even if it's easy to access, we should always remember that you need a certain expertise because this expertise, the one that we have at Lexogen, really remains fundamental in order to get relevant outcome. And making things easier led people to think that they can do everything in a simple way. And we are here to help them understanding that, no, you need first to understand exactly what you want to achieve, and you need to have the right expertise. And to conclude on your question, what inspires me most is the fact that we still are at the beginning of the understanding of the full potential of the RNA sequencing outside of the academic world, because we start to see it in drug discovery, in diagnostics, and also in personalized medicine. And this is really the future. And now with AI coming into the picture, we are not just looking at the RNA anymore. We are really starting to deeply understand what really drives human health and disease. And this is the beauty of using RNA sequencing.
I'm going to pick up on that last point, that AI point, because there's something that's a bit of a cliche in biotech now, which is that AI is only as good as the data. And I guess both of you see that in practice. But Quinn, what does it mean in concrete terms to design data for AI? How different is that from how data has historically been generated in the field?
Well, just to echo the obvious, right? An AI model is two things. It's when a good algorithm is matched with good data, and you need the both of those for that to happen. And unfortunately, what's happening in a lot of the AI hype at the moment is a lot of focus on algorithm. And in certain areas, it's done very, very well. The large language models have done extremely well because they've had a ton of data to train on. We don't have that advantage in biology. We need to go away and generate good data. And that's where the real challenge comes in. So I think for me that when we talk about what is good data, there really are two things. One is, I think, and this is something we think a lot about at Ochre, is what is the type of data I need to answer the question? So if we're talking about discovery data, it really needs to move beyond just big correlations. I think one of the big challenges, something that people don't fully maybe necessarily appreciate about AI is just a very clever way, for the most part, to detect correlations that the human brain can see. It's just correlations. It's nothing else. And so it doesn't address the big questions in biology when you're doing discovery. It doesn't address complexity, so all the complex interactions that play out in biology necessarily, and it doesn't address causality. In other words, if I change this gene or this protein, it affects this, and it has this consequence on the patient. So you have to go and actually generate the data to allow the algorithm to more explicitly address those questions of complexity, causality. And so complexity, for example, we address that by running very complex human models. We culture diseased liver tissue out here in Asia. We have these organ perfusion systems in New York where we're keeping whole human livers alive to sort of generate the data to study what's going on then. And then together with sort of Lexogen, we have generated a lot of causal data where we've gone and knocked out all the genes in multiple human liver donors, disease states, RNA sequence to build up this fundamental wiring diagram. The second part of the good data question is sort of implicit in all of that. As you can guess, all of that is very, very, very tough data to generate. I mentioned all primary human work, complex human tissues, models, big screening studies in primary human cells. Getting the right level of data at scale that is high quality, by that I mean reproducible, etc., is extremely challenging. And I think that's why we're very proud of this project that we pulled off together with Lexogen. It was to an incredibly high standard.
Stefan, if I can just follow up on that, what role do the specialized NGS providers like Lexogen play in this development that Quinn's talking about?
Yeah, first of all, I would really highlight that I fully agree with Quinn's previous comments. And today, standard sequencing run is actually not enough anymore. If you want to use AI in a meaningful way, you need data that is designed very carefully for a specific question. And this is where Lexogen can make the difference. It's true that RNA data is very rich. It gives you much deeper view than traditional tests, which is very powerful for the complex diseases, as mentioned by Quinn before. But this means also that you must generate this data in a very controlled and very careful way. Otherwise, it loses value. So our role at Lexogen is really to make sure that the data we produce is clean, consistent, and useful for our partners. So you can think somehow of this data like the fuel for AI. If the fuel is not good, the engine will not work well. So the project with Ochre is is really a great example, because if you want to lead in AI, you cannot rely on old or already public data, like unfortunately we see frequently. No, in this case, you really need to start with the right data from the very beginning. And for sure, this has a cost. It's clear. But on the other hand, sequencing is becoming more accessible, faster, and also more flexible. So this is allowing us, at least at the level of Lexogen, to support more and more very large projects today that were not possible in the past, given the cost. And at the end, I would say that even if it's an investment, it's really worth it, because such kind of project can really improve diagnosis and can help more targeted treatment for patients. So it has a cost, but the return on investment is really huge.
Now, it's been a little while since I've had two guests on the podcast at the same time, but I think in this case it makes a lot of sense, because this partnership and this common project that you're talking about, that's what I really want to dive into today. So Quinn, tell me, how was it that Lexogen and Ochre came to work together in the first place?
Well, the marketing jingle, "Come for Rex, stay for a while." I think it was a little bit like that with Ochre and Lexogen. The sort of I first got to know about Lexogen during my academic work in Oxford, and came for the great technology. The chemistry is fantastic, and it really performed very well compared with a lot of the other things we were using in-house. But it's really when we started thinking about, as a company, who we'd like to work with, that was when the stay factor came in. And it's not just this perturbation project, it's the big spatial sequencing project that we did together with Lexogen in the very, very early days. In all of these projects, the stay factor was really driven by the fact that Lexogen is a team that always delivers. And I know Stefan knows this because I say this all the time. It's why we keep coming back. This team really has a culture of doubling down, finding a way to get around problems, getting to solutions, working very closely with us as a team, and then always delivering gold. It's very difficult to find that kind of level of service and dedication from a team.
Quinn, we're talking about one of the world's largest human liver functional genomics data sets. This is clearly not a small undertaking. What does a project of that scale require at the strategic level? And why was a dedicated specialist partner essential rather than just going with a standard service provider?
I think for us, the best way to summarize this is as follows. This data set that we generated, where we knocked out all expressed genes and multiple donors and disease states, is a core foundational data set for us. So you'll have heard in AI, we talk about foundational models and then disease-specific or domain-specific models built on top of that. We have the same sort of workflow in Ochre. And this data set we generated with Lexogen is a foundational model that we're using to make predictions around a lot of programs for the next 2, possibly even 3 years. And so it absolutely had to be top-notch and high quality. We really, really couldn't skimp on this one.
Stefan, on your side, handling such a large project with that sort of consistency that they need for the AI applications, that's not straightforward either. From your side, what did it actually take to deliver at that sort of scale, and what sort of critical decisions were there when you were designing that workflow?
So, first of all, I would say that such collaboration is not only a technical adventure. It's also a human adventure, and not only to have a good dinner with Quinn in Vienna. No, it's really a human adventure, because for a project of this size, I would say one of the largest in the world so far, alignment and mutual trust between the teams were really essential. So it was not only an alignment on the technology side, but also on the way we work together. And we had really a very open and constant communication with the Ochre team to make sure that the data would fit their AI models perfectly. And this open discussion was really key for success. And one important choice that we had to make was the technology. And we have decided to use our TOR technology, which is a method which doesn't require RNA extraction step. And this has helped us reducing the variation. And this has made the process also faster and more reliable. In addition, this technology is very sensitive because you can detect gene expressed at a very low level in a cell, even with less sequencing depth. So this was an innovation that we ran a year, 2 years ago, and I'm glad that we have been able to demonstrate the power of this technology with Ochre, and that they were trusting us. But this has made at the end the project both efficient and high quality. Also, to ensure the success of the project, we have performed several pilot experiments in order to fine-tune the parameters on both sides, but also to test the robustness, the sensitivity, and the reproducibility of the workflow with the hepatocyte, with the specific Ochre starting material. Sorry, again, my French accent. So we also produced a unique batch of reagents. Like that, we have avoided variability. And last but not least, we have set up very high quality standards, and we have worked in a blinded way. So it means that at Lexogen, we didn't know what kind or which samples we were processing. And Ochre, on their side, they kept that information, and later they checked the results. And in the end, we have been able to reach the 95% success rate that they were expecting, which was a strong confirmation for both teams, I believe. And I would like to take advantage of this discussion also to say that really people are key to the success here. And I want to congratulate and really to warmly thank all the members of the team which have participated to this achievement. They have been fully dedicated. They have contributed to a very high level. So yes, a huge kudos and huge thanks to everyone on both sides.
It seems like incredible alignment. Quinn, can you maybe walk me through the outcome there? Why was such high quality, consistent data just so crucial for your model?
Well, so as mentioned, we think of this as the foundation, a very big foundational data set for us going forward over the next 2 to 3 years. And we do, without getting too technical, we do 3 things. And are doing 3 things, we'll be doing 3 things with this data over the next few years. One is this data set, just at a more basic biology level, allows us to directly model what's happening with genes. If you go ahead and knock out every single gene, in different donors and different disease states and RNA sequence, it gives you a wiring diagram, a fundamental gene regulatory network that you can go away and think about. And so we, for example, have been able to very quickly ask, using these sort of approaches, ask questions like, what happens if, which genes in hepatocytes could upregulate FGF21? This is a very important protein at the moment in the liver fibrosis space, and the current therapeutic strategy is just to give the protein as a therapy. Another approach could be actually to upregulate it from within the hepatocytes. And so using that approach has just been really instrumental in helping us accelerate in what is a new exciting therapeutic class. We can also, a second thing we can do is look at how this foundational model, this disease set, this data set modulates diseases. So alongside the work that we do with Lexogen, we're also, for example, single-cell sequencing fibrotic donors all over the world, in Asia, Europe, and the US, and we can look at how this data modulates those signatures. And we're using that sort of approach to come up with new ideas around fibrosis biology and propose therapies that we're then sort of validating in our models. But I think the really interesting AI story in all of this is the third approach that we do that. Now, there's this term in our space called lab in the loop, and I'm always very nervous about it because I think teams that often, it's similar to the adage that teams who talk about AI are often not doing AI, and the teams who are not talking about AI are probably the ones doing AI. It's a little bit like that with lab in the loop too, I think. It's a very hyped term, but I think as an idea, it's very valuable, and we do it at Oak. And here's what we do, and here's how the data that we generated with Lexogen enables that. So I mentioned complexity right at the very beginning of this chat. We are obsessed with complexity. We do not believe that you can appropriately model very complex processes like fibrosis in very simple cell culture models or inappropriate animal models. And so we work in human tissues and whole human livers. Now, you can imagine that doesn't necessarily scale very well. We can't go away and brute force test every single gene. What we can do, however, is we can go and perturb 1,000, 2,000 genes in our model. That is a scale in our models. That's a scale that we can do. And then we can train an AI model on the causal data that we generated with Lexogen, using the output from the 1,000, 2,000 genes to predict the other untested genes. And so that allows us to, in silico, using AI, scale our predictions using a very complex model. So we don't have to go back to cell lines, very simple culture. We can use these types of data and AI to really scale. And I honestly think, that's without getting into, again, the hype of lab in the loop and how we do these kind of techniques. I really think there's merit in this sort of approach, and this data set has enabled us to do it.
Quinn, when I was getting started in business, 30 years ago, I was hiring for my first teams. I had to do my first reference checks on people, and I had a whole list of questions that I wanted to go away and ask. Another manager who was a bit of a mentor to me said, there's really only one thing you need to ask, which is, if you had the chance, would you work with that person again or not? Let me ask you the same question, though I think I can understand the answer based on your enthusiasm. Would you work with Lexogen again?
Absolutely, in a heartbeat. And it's not just for the return factor, the ability to just really, really deliver all the time. Something that I've never told Stefan this, but it's something that I also just deeply admire about the team is their real intentionality in their leadership style and how they do things. If you walk into the Lexogen labs, and of course we've been around to visit a few times, there's just so many things, so many ideas that actually I've stolen one or two of them, right down to how Stefan builds the organogram upside down to demonstrate where leadership sits supporting everyone else rather than the more traditional organograms. They just think very differently about things as a team. And I think that really does play out in their culture and how they deliver with their science.
Well, Stefan, you can take that straight back to the team. But let me ask you, how do you think about Lexogen's role in this broader shift towards AI-driven, human data-led drug discovery? Is this a new kind of relationship between sequencing providers like yourselves and biotechs?
Well, first of all, let me thank Quinn for his kind words. Thank you very much. Well, actually, I think that for companies like Ochre, choosing a sequencing partner is really a strategic decision. And I don't think it's only about outsourcing work, and this is also what was said somehow in substance, Quinn, earlier. But it's also about finding a partner who understands the science and can adapt to your needs, and where you can work in full trust, because you are sharing some values. And we see ourselves here at Lexogen as part of our partners team. We see ourselves an extension of our partners team. And we are often involved very early in the processes, which is helping us, helping better our partners with the experiments, so the data will be meaningful for them later on. So this is something that we really think about. We want to be the extension of our partners. Then at Lexogen, as I said earlier, we are very specialized because we have 20 years of experience in RNA sequencing solutions only. So this may sound a niche market, but this is actually our strength, because we have now a bunch of experience, and thanks to this, we can really adjust our workflows and work with many different types of samples, even the most difficult and challenging ones. So I think that this is one of the key strengths of Lexogen. And at the end of the day, the good decisions frequently depend on good data. We said that earlier, and I want to re-emphasize this message. But this is why the team is really focused on how we can bring more consistency, more quality, and control at every step. We also are very focused on data security and IP. We take that very seriously so that our partners can work with us in full confidence. And I think that this is also something very important, especially when you have data which are very sensitive. And I would say that finally, speed in the project, for instance, was important. But it's only important the speed if you have the good quality. Otherwise, it's not so good. So delivering a project in this scale in 5 months was not just about speed in our case. It was more about doing it right. And the whole team was behind this. And I think that this shows what the team is capable of.
Let me zoom back out again, because I think the implications of what you've been talking about with this project go well beyond this particular project. Quint, do you think this broader drug discovery industry are we genuinely shifting into this human-first, AI-enabled model, or is it still pretty early days and most everybody is still watching to see what's going to happen?
Well, I'll speak mostly for the liver space, and I'd say the intent is there, but we are at the beginning of the beginning. So what excites me about what we're doing better with human data in liver science, and particularly the translation as we get towards clinic. So, sure, regulators and particularly the FDA are leaning more into human models. You do not have to do animal studies anymore to do your first in human. According to the FDA. The reality is very different right now, but the intent is there. And so we can work with that. And of course, at Ochre, we think about how our model data can play into that. We can model biomarkers in upper fused livers and use those biomarkers to inform our clinical strategy. On the topic of biomarkers, that's another thing that I think is we're getting better and better at, moving away from traditional endpoints to more biomarker-driven discovery. And the FDA recently in the liver space has finally said you probably don't actually need to rely on biopsies that much in clinical trials. Now, that's a bold move, and now we're sort of moving to biomarkers of human endpoints. But nevertheless, I think it's a very, very positive move, and that will enable us to think again about more complex analytics and where we want to go and the kind of predictions we can make from that. And then, I mean, again, and this has all happened in sort of the last year, the FDA has really leaned into more complex clinical design. So FDA, Bayesian style design doesn't have to be AI, just even Bayesian style design so that you can borrow data from previous studies, you can borrow data across arms, is really enabling us to think, I think, more ethically and in a much more sophisticated way around human data, of which AI is part of that. So I think the pieces, so that was a very long answer to say, I think, at least with the liver space, and I think for a lot of the clinical trial space and the therapeutic space, the individual pieces are there, but we're on a journey. We're not there yet.
Well, if that's the bigger industry space, then where does Ochre go from here? I mean, you've got one of the largest human liver functional genomics resources that's around. What does this foundation make possible for your pipeline? What does it lead to for people with liver disease?
Yeah, great question. Yeah, as I mentioned, as I hinted at in a few comments ago, the beauty of having big foundational data sets like the one we generated with Lexogen is that it allows you actually to ask a lot of questions. So as a company, we're very, very focused on fibrosis biology right now. So we're talking about cirrhosis as the end stage of fibrosis in liver disease. And the assets that we're pushing into the clinic at the moment are all very much focused on that question. However, we are working with a major pharma partner thinking about liver regeneration, which is really, really, really interesting. That actually might have implications for fibrotic disease, but even the notion of being able to restore liver function by getting livers to better regenerate, I think is an incredible thing to be doing and something that we're doing together with them. And we can ask questions about how the liver impacts the rest of the body. We have another pharma partnership where we're now looking at liver targets that impact obesity, really thinking about liver-directed therapies rather than appetite suppression therapies and what the impact could be in terms of muscle sparing. I think everybody's heard that Wegovy makes you lose a lot of muscle mass too. So what could we do there? And also in terms of improving the antifibrotic response. You lose weight, your antifibrotic response gets better when we're talking about certain elements of liver disease. So we're able to really, it's almost like being a kid in a candy shop sometimes, and you've got to learn to focus because data like this allows you to really deep think some fantastic questions. And we're just going to keep pushing on those for the next few years.
And Stefan, on your side, where's Lexogen heading? This project really set a benchmark for what's possible with sequencing when it's designed for AI. What does the next chapter look like for Lexogen's service and their technology platform?
So looking ahead, I would say that I really believe that collaboration like this one will become kind of new standard because they really show what is possible when data is created with AI in mind from the beginning. Now, what I really appreciate also with Ochre is that there is this ethical approach, trying to work less with animals and more with human tissues. And that said, if we want to expand that, because I believe that this kind of approach will allow better diagnosis and treatment, but not only with liver diseases, but with other organs and diseases, I think that with some tissues, we will not have a lot of cells. And what we have been able to demonstrate also is that with our TOR technology, you can start with also a very limited amount of cells. So with that in mind, I really think that this technology will allow us to do this kind of big projects, but also starting with a very tiny amount of material. And I think that this is the future. Now, to be honest, the goal of our team is just to use our expertise in RNA sequencing to help innovative companies like Ochre, turning scientific discoveries into real medical progress. And I must admit that even if we do not take credit for the Obelisc project, which is really a remarkable innovation which has been developed by Ochre, the whole team at Lexogen is really truly proud to have played a part in something that can help somehow in the future patients' lives. And we are a technology enabler, but when we see that there is a science behind what we are doing, when we see that we can help these companies like Ochre succeeding in helping improving the life of patients, for us, it's worth everything.
You know, it's always fascinating to hear how one of these partnerships works behind the scenes and all of the pride from both sides that goes into it. So thank you both for this conversation today. Quinn, if people are interested in learning more about what Ochre Bio is doing, your mission, your work in liver disease, where can they go?
It's very simple. Website, LinkedIn. Check it out. Thank you.
And Stefan, if people want to learn a little bit more about Lexogen and what you have to offer, where can they go?
Here we are not very innovative. So it's also the website. It's also LinkedIn, but we have X in addition.
Perfect. Stefan, Quinn, thank you both for joining me today here on Beyond Biotech.
Thank you, Dylan. Thank you, Quinn.
It's been an absolute pleasure, guys. Thank you so much.
That's it for this episode. Don't forget to check out all the latest news and analysis at Labiotech.eu. I hope wherever you are in the world, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.